All right, so our next speaker is Sarah Lynn, who comes to us from Edmonds College, where they studied cannibalism in insects. So we're excited. Sarah Lynn's on their way to University of Washington and has tackled this really cool question of how all these many restriction factors that exist in our innate immune system might work together to combat pathogens. So Sarah Lynn.
Hello, my name is Sarah Lynn Ferrero. That was weird, sorry. I completed my internship with Amanda Norstein as my mentor in the McLaughlin lab. And my project was on how two proteins from the APOBEC family might interact with each other in restriction of LINE-1. So I just want to go over some background. My lovely lab mates have already kind of talked about this, so I'm just going to speed through some stuff real quick and just talking about how L1s or LINE-1s are the only retro transpositions that are currently actively dividing in the human genome, that they use the body's own system, its own polymerase, to make a copy of its own RNA, which then encodes for the proteins, which then attach themselves to the L1, shuttle back into the nucleus, nick a portion of the genome, and reverse transcribe. This mechanism creates two copies, one at the original site and now a completely different one, somewhere away from its original location. And this itself has measurable negative effects when LINE-1 is able to replicate unchecked, especially in terms of its seen increased expression in cancers, autoimmune disorders, and certain neurodegenerative diseases. So I kind of want to talk about the amount of restriction factors that are in your body at any given time. And when I say several, I really don't think I can understate how many. Every single little circle or possible just word over here is a restriction factor that at any given time is attempting to keep LINE-1s from replicating. And I am currently focusing on one teeny tiny little family. So when we're trying to talk about how in research you usually isolate down to one specific protein, you're kind of losing the context of how all these things are interacting with each other, especially in the fact that previous research has shown that APOBECs specifically have the ability to form multi-protein complexes. So they do even interact with each other. So how might these restriction factors be influencing each other? That is my question. And obviously I can't look at every single restriction factor because that's just too much. So we focus down to APOBEC, and specifically from there, that family is just way too large. There are 7 members. So picking 2 specifically, because obviously as you increase the size, you increase the number, you increase the complexity, and A and B especially are considered potent inhibitors. So I'm kind of just going to go through their mechanism and the fact that they do what's called cytosine deamination. So they will affix to transiently exposed single-stranded DNA in the sense that when LINE-1 replicates, it replicates with R1, and there's this belief that there is a deaminase independent mechanism where there's a high affinity for that RNA, which will then cause the APOBEC proteins to bind to the RNA and shuttle it out to be degraded. So now you have this transiently exposed single-stranded DNA, which, because LINE-1 is entirely dependent on humans and our mechanisms to be able to copy itself, it will have long periods of time where it's just a single-stranded DNA, and APOBECs specifically go after those. So they will attach, hypermutate from cytosine to uracil in such extreme measures that a protein will come behind, see that uracil is in place of cytosine, which is not supposed to happen in DNA, and will excise that portion of DNA and will then send it off to be degraded. That, of course, is the best case scenario, where line-1 is completely removed from the genome after while it's attempting to transcribe or just after it has been transcribed. But there are some cases where it doesn't mutate enough, and then you have repair. But because it's still such a high level of mutation, those proteins that are critical for line-1 to be able to be shuttled back into the nucleus and replicated no longer work. So this is basically its last attempt at retrotransposition. So for us to even be able to choose A3A and A3B as they're known, because I don't want to keep saying APOBEC, it's too much. Are they even together? On a single cellular level, A3B is completely inside the nucleus. A3A is found both in the cytoplasm and in the nucleus, so we're seeing them in similar places in an individual cell. When we're talking about tissue, specifically just these ones that I've left colored, both in lymphocyte tissues and in a whole array, they are expressed together. So they do even have the possibility to interact. I also want to acknowledge that why we even decided to do this research, because it is kind of it can feel a little random, but a PhD candidate at the Alvarez lab for Fred Hutch is working on how APOBEC might be implicated in cancer, and has found that when A3A and A3B are expressed together, their observed deamination specifically is much higher than the expected deamination when they are together. So they're doing something that is additive beyond what we would initially expect. Obviously, the scope is more for cancer research. So we're taking that and saying, can this same principle be applied to line-1? This is where I kind of want to get into my methods, where I'm asking this question: when multiple APOBECs are present, how might this impact the restriction of line-1? So the first set is just trying to do them together to be able to get a baseline for how they work on their own, to then build off of that for my next two questions, which is how do these combinations then restrict together? And then how is there possibly any combination interactions outside of their deaminase activity? So in their deaminase independent interactions, are they also helping each other? So the thing I basically used every single week, dual luciferase assays, wonderfully completing them every single day. So you take your line-1 plasmid that you want and you put it into your 293 T cells along with varying concentrations of your APOBEC3s, either solo or combinations, depending on what stage you are in your project and what question you are trying to currently answer. So you're putting them together into your wonderful 96-well plate. You're allowing for 6 days of growth, and then you are taking them to a machine to measure your firefly luciferase, which Amelia kind of explained, where the luciferase is coded into the line-1 plasmid, and it's reporting line-1 activity. For every replication, you get another luciferase protein. So as activity increases, you would expect your luminosity to increase. So it's specifically looking at line-1 activity. And then you're repeating this again with your Ranella, specifically looking at cell health in terms of how many cells were able to take up your transfection plasmids. And because even at extremely low concentrations, A3A especially is highly toxic. So you might come out of it and say, oh my gosh, A3A, you were able to wonderfully restrict L1 activity. There's no stuff left. And then you're like, oh no, my Ranella is telling me that you didn't just stop line-1, you also just killed all my cells too, which I guess that works, but not for my experiments. So coming out of this, my first step was those response curves. In this idea that, as you're seeing here for A3A, we are getting into 0.5 and less, where I'm just trying to get my cells to stay alive, but still inhibit in a range where, as I look at different percentages, I can actually see how this restriction is going. So I'm not just going like, oh, I'll put 75% restriction for A3A and another 50 for A3B, and now everything's suddenly all blown out and I can't see the variance between it. So it allows for precise concentrations for the expected restriction. Here's the wonderful little formula that I had spent way too long trying to figure out. And obviously the same thing was done with A3B to then get me into my second part, which is my results and analysis, where I did my wild type combinations, where you would have a certain amount of A3A or A3B, and what you obviously, in relation to that amount, what you expected to be restricted. And this is basically just trying to show that this one didn't come up with any conclusive results, in the sense that you would expect, as you increase, the amount, which is going from here to here, you would expect a decrease in activity as more restriction factors are available. And it's just all over the place. So unfortunately, this one just didn't come out. And at this point, it might not really be showing anything unless we can kind of, yeah. So also, I probably should explain that these are done in ratios and not just simple, and not just like the whole luminosity, again, trying to account for the fact that cell death is very much possibility in this. So when the ratios are above 1, you're looking at high L1 activity or the possibility of just a ton of cell death. And at lower ones, you're looking at low L1 activity and extremely healthy cells. So you're wanting to see that as you're increasing concentration, your ratio is dipping below 1, because you're getting to a point where there's not a lot of L1 activity, but you do have really healthy cells. So when looking at my wild type and catalytically dead combinations, catalytically dead pointing to the fact that those proteins cannot interact with deamination. So their only way of interacting is independently of that deamination mechanism. So they can only either be coupling together with the wild type protein, or in basically just assisting, not taking part of the main interaction between these proteins and LINE-1. And this is where things actually get really interesting, in the sense that this is what you would expect your wild type A3B by itself to be. When you add catalytically dead A3A that cannot deaminate but can assist, you are seeing measurable decreases, and especially that as you increase your catalytically dead, you are also seeing decreases in activity, which is kind of interesting. It really is. There's unfortunately like a flip with this, where what we're seeing instead is catalytically dead B, as you increase its concentration with wild type A, it actually is reducing, seems to be reducing wild type A3A's ability to restrict instead. So kind of just reiterating that increasing catalytically dead with wild type A3B seems to decrease the effectiveness of restriction, whereas adding catalytically dead A3A has a more expected pattern. So kind of going back to what was my experimental question and what were the outcomes for this, I was able to create a dose response curve that facilitated further experiments. So yay, that worked. Wild type combinations didn't necessarily show any conclusive trends, but catalytically dead can both inhibit with wild type A3A and catalytically dead A3B combos, or assist, as what as catalytically dead A3B was doing with wild type, or catalytically dead A3A was doing with wild type A3B, depending on what your protein combinations are. And kind of some ideas for future work, looking at other proteins in the APOBEC family. Again, there are 7 others. Trying to repeat my experiments with the wild type combinations, just because they didn't necessarily work, but it could definitely be how my experiments were set up. Can other restriction factors beyond the APOBEC family in combination with A3A increase its ability to restrict, but allow it to be done at lower doses, so you reduce that toxicity if you're looking at therapies? Can this be scaled up to more restriction factors outside of the family I've chosen? Can this be put back to Cheyenne's work in the Alvarez lab and Fred Hutch, and how APOBECs might be influencing tumor genesis? So, there's tons of questions, especially in the immediate, but also in future ideas. I would like to thank all of the wonderful mentors that I had at the M'Laughlin Lab: Rick, Amanda, Ricky, Joey, and Lay. I would love to thank Cheyenne for providing me with these catalytically dead samples that got me such wonderful results. And I would love to thank the Pacific Northwest Research Institute for providing me with this opportunity. Do I have any questions?
Amy?
The catalytically dead mutants that inhibited activity, so
Mhm.
we would call that a dominant negative
Mhm.
effect.
Yeah.
Are there any cancer mutations, or I guess there are cancer mutations because that's where you got them, but are there people who have diseases associated with any of these catalytically dead mutants that have some kind of phenotype?
If they do, it is not something that I saw in the literature. The unfortunate thing is, and I don't know if you noticed, but a lot of the information that we understand right now of how APEC works has happened within the last 5 to 6 years. So we're really in this place where we're saying, we think this is how deamination works. We think these are their independent mechanisms, but we're still trying to figure that out.
I think the nice implication of your work could be that the prediction would be that the inheritance pattern in a human would be that this would be an autosomal, presumably autosomal dominant disease.
And it might happen somatically too, which would be cool to look at in cancers. Right?
So Amelia also worked on restriction factors.
Mhm.
in MOB10.
Yeah.
And MOB10, a mutation in MOB10's ability to combat violence can also lead to problems, right?
Mhm.
Do you see in your literature and work something similar? Because I think I saw a graph earlier that was like APOBEC3 and APOBEC3A and B work separately, but together they inhibit better. So
Yeah.
if one of them is mutated, can the other one counter it, or?
Yeah, see, that's the thing with especially APOBEC. This family has had a huge expansion in the sense that, like, mice models have only 1 APOBEC. Humans have 7. And we're seeing that this huge radiation in the family has allowed for a significant decrease in LINE-1 restriction in humans in comparative to mice models. And a lot of that is also because the APOBECs aren't all necessarily localized to the nucleus. Some of them can only be in the cytoplasm. So they're working on restricting different areas of that LINE-1 pathway, where A3A and A3B especially are working on that last step. But as Amelia pointed out, like MOB10 is in areas where it's inhibiting restriction straight from the start, or even working on degradation in the cytoplasm. So it is a lot of like forced redundancy to reduce LINE-1 activity, where you have all those myriad of restriction factors all trying to keep this down because of its negative effects. Mm-hmm.
Thanks, Sarah Lynn. All right, only 10 more talks. All right, our very last talk, unfortunately, couldn't be here in